So the first presentation will be given by Pierre Gratier from Bordeaux about deriving physical parameters from molecular emission.
Thank you. Okay. So yes, I'm going to talk about what Jérôme alluded at the beginning is it possible, can we derive physical parameters directly from a molecular emission? But implied here is without using radiative transfer models. From the structure of the molecular emission and the relationship between the different lines, is there a way to recover some physical parameters? Why is it important? Well, high resolution, I mean, in the range of 30 to 40 arc-second column density maps, they come usually from just emission maps in the far infrared or millimeter, for example, Herschel, or star count and extinction. And what has not been mapped by Herschel won't be mapped in far infrared for some time, because we don't have any more far infrared telescopes in the sky. And the question is, can we use molecular emission for one particular physical parameter, which is column density maps? And also, what about other quantities, T-dust or other physical parameters? In 2017, we made a first paper, which was a qualitative paper, using a principal component analysis on the data set that you've been shown, so several lines across a large map of Orion B. And to see, the main question was to see how the emission from molecules were related to each other, which ones were correlated together, and which one were anti-correlated, and which one did not depend on each other on a linear scale using a principal component analysis. And what we found is that after doing this analysis, the principal components themselves were correlated with physical quantities. For example, NH2, you see here, the principal component was correlated with NH2, but we had smaller but still present correlation with other physical parameters. So it seemed that combining the molecules in a certain way could give information about physical quantities. And we made, for example, one map like that, which could give a synthetic view of Orion B. Encoded here, luminosity is the column density. The brighter, the more column density, well, the more value in the PC1 quantity. And we could encode in color here two other densities and see, for example, the UV illuminated but not too dense parts in orange, the non-UV mid density in light blue, and the very dense and non-illuminated parts in blue, for example. So we had a synthetic view just from the principal component analysis of the whole structure. And then the question was, how can we go from qualitative to quantitative? Can we get a number of NH2 if we get a value for different molecular emissions? So the idea was, how do we go from this to this? And is it possible to do it? So that's what we did in a paper a few years ago, and that I'm going to focus now for the rest of the presentation. And to do that, we use the methods of supervised machine learning, which are very general. And the idea is to learn a relationship between two data sets. So the first one data set is going to be the molecules, and the other one is going to be one physical parameter, here the column density. So we want to learn that. For example, linear regression is some kind of supervised machine learning. You learn the coefficients, and then you can predict a quantity from the abscissa or the input variable. The predicted quantity is what we want to learn, so the NH2 column density. What we'll call features is what we want to learn from. And here's the integrated intensity and peak temperature maps for 18 molecular transitions. So we had a total of 36 features to learn one thing. In this method, it's very important to put a set aside, what we call a test set, that we are not going to learn from. We're going to learn from everything except the test set, and we're going to test at the end the capacity of the method to predict correctly the values from this. So here we selected this, which is the Horsehead part of the Orion B that we knew well from before as the test set, and we learned from everything else, but the learning part never sees the Horsehead. And so what algorithm did we use? We used the random forest regression algorithm, because on a scale from less complex to more complex and less interpretable to more interpretable, going from very simple linear models to a very complex neural network, was somewhere in the middle, where we felt it would be complex enough to get the right prediction accuracy, but still interpretable in terms of chemistry and what we already know about the interstellar medium. Okay, just rapidly, decision trees is just, for example, you want to know what fruit you have, and you know some parameters of the fruit that you can measure, for example, color, size, or shape. And every time to write to an answer, you ask one question on one parameter. And then at the second level, once you've decided what is the color, you can decide what is the size. And this is for classification, but it works the same way for regression. I don't have time to go a lot of detail into this, but you have a GitHub tutorial here that I did a few years ago that explains decision trees, and then what is a random forest, which is a group of trees to increase the accuracy of the method. And so here are the results. This is what is observed, so that is what that has not been seen as a column density, but we want to check how it is reconstructed. And this is the prediction from the 18 different molecules. And you see here this difference map, and you see that it's quite flat. So you don't seem to have a lot of bias on the reconstruction of the column density. Quantitatively, we can reconstruct the column density within a factor of 20%. Well, column density, reconstruct the Herschel maps within about 20%, with a maximum error here about 80%. And you see here that although there's a slight bias here, it's mostly compatible here within a factor few within a few tens of percent of the observed and the predicted values. And then we thought about, okay, we have a predictor now, but we're not going to stop here. We're going to try to understand how it came to that result. And for that, there are many standard ways that actually could be used with almost any supervised learning method. And one of the main questions is to ask, how did we get that result? What are the molecules that are actually important? And are there some that don't matter a lot to get the result at the end? And to do that, for example, what you can do is you can train with replacing one of the features by just noise. So you don't add information, you remove part of the information by replacing one of the features by noise, and you check how worse the result is. And you can order them by feature importance. And for example, if you replace the 13CO map by pure noise, you lose a lot of predictive power because 13CO is important to get the full column density. So this is the order of the different molecules. The usual suspects are at the top, 13CO, 12CO, C18O, but HCO+, HNC, and CCH are still important to derive. So this is computed for the whole map. I don't have time to talk about it, but I have additional slides. We also showed that the molecules that are important depend on the regime of the interstellar medium. In denser part or more diffuse part, it's not the same molecules that are important. This is an average of all the whole conditions in the interstellar medium. You can also have a look at another way. If you retake these molecules, have you seen which ones are the most important, and you add them one by one. So you try to first do a prediction with only 13CO. So it's possible to do it, but it's not the best way to do it. You see here that you are here. If you add 12CO, you reduce a little bit the root mean square error, and you reach kind of a plateau here where additional lines, on average for the whole range of conditions, do not help you a lot to get new to reduce your uncertainty on the prediction here. What we could also do with random forest is to do what is called contribution map. So for each pixel, we ask what is the contribution of each species around the mean log NH2. So the trunk of the tree of the random forest tree is the average value of NH2. And for each pixel, each of the different questions that you ask will add or remove from this mean value. And you can construct a contribution map saying that, for example, for pixels around here, there's a very strong positive value of 13CO that add to the column density or to the mean column density. And from that here, we find that it's compatible with what we know of the interstellar medium and the chemistry and physics of the interstellar medium. The 13CO traces most of the column density of gas. 12CO starts to be, is not very important actually where 13CO was important. It's near 0 here. Well, it's because it's optically thick, so it's not measuring the column density. It's measuring something else, probably the temperature. You only see the surface, but when it becomes less optically thick, it starts to have an importance. And it's important complementary to 13CO because 13CO, you don't see it anymore. And so it takes the next step to have the column density in regions where 13CO is not here anymore. C18O is the opposite for the most dense parts. And HCO plus is important to trace the UV illuminated region. So in Orion, the star is here and illuminates that region. So without HCO plus, you cannot reconstruct well the UV illuminated regions, and you need it for these regions, but it doesn't appear to be important for the rest of them. What you have to be careful with regression method is to be beware of extrapolation. So most supervised learning are kind of fancy interpolators. They do fancy interpolations in a parameter space, but they break down very ungracefully when predicting outside of the subspace of the training set. And the subspace of the training set is not a hyper rectangle. It's the data itself lives in a subspace that you cannot reach. We know, for example, in the interstellar medium, there are no places that are both diffuse and cold. I mean, the diffuse part is hot. And so even if you try to make it predict in that region, it will say something without letting you know that it's completely wrong. And so what we did is that we tried to model the distribution of our parameter space with the Gaussian mixture model, which enabled us to quantify, if you have a new data, is it similar to your training set or not? And from that, you can do a first step. Before trying to do a prediction, you test if it's indeed similar or not to the training data. If it is, you can be quite confident that your prediction will be correct. If not, you should stop and not try to extrapolate. You should get more data on different regions to populate this region of the parameter space that you've never trained on before. Of course, I've told you you should not extrapolate, but I'm going to do it anyway. We've checked that indeed, when doing this extrapolation, we were still within the range of what was trained on. And we did two tests. We did the first test to add noise to the data set and to see how it breaks down if your data set is not as sensitive as the Orion B data we have. So you see here that as noise is added logarithmically, so there's a lot of noise here, it breaks down around a few Kelvin of noise, which is quite a lot. It's still quite good to up a few Kelvin of noise. And on the right, what we did is that we tried to see how far we could push Orion B and move it away, so by convolution and downsampling, to see if we could still recover. If the information at high resolution was enough to recover low resolution. And you see same thing here for RMSE. It breaks down if you push Orion B 4 or 8 times farther, but it doesn't seem to work for farther places. Finish with future plans. Can we do it for other quantities? For example, G0. Well, no, not with the method we did, with the conclusion of the paper we arrived. But more generally, Luca Henning is working on looking at mutual information between molecular and physical parameters to see if this is a problem of the method we use, or if it's a problem that there's no mutual information between G0 and the molecule that we choose. And possibly, one solution, if indeed there's information, we need to find a method that can recover it. If it's not, we'll have to find what molecule can actually trace. Maybe it's because we only have one transition of each molecule. Maybe if we had the different temperatures, it would be different. Thank you.
Thank you, Pierre. You have questions, Pierre? Those are plenty of questions. 9, maybe?
Well, did you experiment this on just one field or on the entire field and looking?
So at the time, we didn't have the right the left part of Orion B. So we did not train it on this part of Orion B, but indeed it's something that we could check now, that if we train it on half of the map, can we test it on the other side of the map? Especially since we know that it's not really the same conditions here. So the question behind it is that, are some of the conditions here not sampled somewhere in the distribution of parameters here? I guess that way should be okay, because here is both PDRs and cold medium. Probably the other way around would not work, because in this region here, there are no UV illuminated, strongly UV illuminated regions. So it's a bit of a question of how far do you need to observe the whole sky to be able to reconstruct something that has already been observed? So there's a compromise here between having to observe all of the Herschel fields, which already exist, and only being able to observe some of them. But part of it is learning what is the shape of the training set and the subspace where the data lives, seeing if new data brings new regions or not.
Very good question.
It's a bit similar. So you use most of this map to train your method. So you would not be able to apply it, let's say, then for Orion A.
Again, this has to be tested. One of the questions is the extrapolation. Is indeed, does it scale to other region? And actually, what is the main question behind is that, is the molecular emission parameter space populated the same way in other regions than this one? If it is, and if the molecules that we have encompass all of the information that is transferred, the answer should be yes. One question, for example, here, we do not have hot corinos or star-forming regions. So there are processes where maybe in other regions that have not been tested here that would break down, but this is something we can check. If the parameter space around the hot cores or hot corinos is not the same as this one, we can flag them before trying to predict. And we can always, one day, add these hot core regions or hot core regions to the training set so that we can be able to test other regions. But indeed, we did not assure that it would be generalizable. On the contrary, we are very prudent to say, be careful when you generalize.
Very good short. What you are showing about generalizing to other clouds. What you're saying is about the different kind of environments, but it doesn't generalize to all the sets of lines, does it? If I give you a line that you didn't train on for another cloud, you can exploit it.
No. But no, I can't exploit it. The other way around, if you have a limited amount of lines that you can observe, I can tell you which one you should start from. Although it's a bit more complicated than just that goes in the paper. It depends on the region you want to. If you look at cold cores, and each entry which is small here actually becomes very important. So this is average on the whole map.
Again,
And see Lucas, yes.
Thank you, Pierre.